If you remember, we talked about the motor neuron and motor units, and each motor unit is capable of connecting to hundreds of muscle fiber or innervating hundreds of muscle fiber. So one nerve fiber and all the muscle fiber it innervates is called a motor unit. So in this picture you see two motor units. Each motor unit innervates hundreds of different muscle fibers. Okay, and why is this important? It is important for the motor unit to be dispersed throughout the muscle to allow the muscle to contract in unison. If not, each muscle fiber will contract individually, and that doesn't make sense. That does not make the process efficient, right? So motor units help the muscle contract in unison by producing weak contractions over a very wide area. Okay? Before we look at the neuromuscular junction, let's look at some important terminology that you'll come across in the neuromuscular junction. The first term is the synapse. Synapse is the point where a nerve meets its target cell. It does not directly touch the target cell, but it is just a point at which it is able to communicate with the target cell. And the junction between the nerve and the muscle is called the neuromuscular junction. Okay, so this will be the synapse between the nerve and the muscle cell. We call it a neuromuscular junction. Keep in mind the nerve and the muscle are not attached to each other. Okay, just keep this in the back of your mind. And each terminal branch of the nerve fiber within the neuromuscular junction form the separate synapse within the muscle fiber. And this is to communicate the action potential or the electrical signals to all the muscle fibers to allow contraction in unison. So if you look at this picture, you see the neuromuscular junction. So this is the nerve ending, and this swollen end is called the synaptic knob. Okay, and right over here is the muscle cell, and you can see that the nerve is not directly attached to the muscle cell. There's a slight gap in between the two, about 20 nanometer in size, in width, and this is called the synaptic cleft. So the synaptic cleft is the gap between the synaptic knob and the sarcolemma, which is the plasma membrane of the muscle cell. Now, around the nerve tissue and around the synaptic knob, there is this blob that you're seeing. Okay, so these are called Schwann cells. Schwann cells basically envelop the neuromuscular junction to prevent the action potential and electrical signals from leaking out during communication. So they basically insulate the neuromuscular junction. So the neuron basically sends the transmission down to the end of the terminal. So it's referred to as a presynaptic cell. So the neuron becomes the presynaptic cell. It's right over here. Neuron is the presynaptic cell. And the cell that receives the signal from the nerve is called the postsynaptic cell. So this cell over here becomes the postsynaptic cell. Now, how does the message get transmitted from the nerve to the muscle? This is where something called as neurotransmitters come in. Neurotransmitters are simply the chemicals that help in transmitting the signal from the nerve to another cell. In this case, it is the muscle. So the neurotransmitters are present in or stored in the presynaptic cell. They are secreted into the synaptic cleft, and when they are secreted into the synaptic cleft, they are able to bind to certain receptors on the surface of the muscle. Okay, so in case of the neuromuscular junction, there is a specific neurotransmitter known as acetylcholine. So the neurotransmitter at the neuromuscular junction is known as acetylcholine. So now over here, this is another picture. It's a blown up picture of the neuromuscular junction. This is the synaptic knob at the muscle fiber. And this is the artist rendition of this image. And the nerve is not directly connected to the muscle. The gap in between the two is called the synaptic cleft. Okay, and these enfoldings are present in the sarcolemma. Why? Why do you need these enfoldings over here? Now, if you thought of increase in surface area, you're correct. Okay, so the sarcolemma has several enfoldings at the neuromuscular junction, and all these enfoldings contain a lot of ion channels to allow the passage of the electric signal from the nerve into the muscle. Okay, and on these enfoldings, you have a whole bunch of ion channels and a lot of receptors that will attach to acetylcholine. Okay, so that's the plasma membrane. This is the synaptic cleft. This is the presynaptic cell, which is the neuron, and this region is the synaptic knob. This over here is the muscle cell. The muscle cell is the postsynaptic cell, and in the muscle cell you see a whole lot of mitochondria, and you see the myofilaments, which are ready to receive the calcium ions for contraction. So acetylcholine is a neurotransmitter and is important for communication between the nerve and the muscle. And for the muscle to receive the communication, they have acetylcholine receptors. When there's a defect in the acetylcholine receptors, it leads to a condition known as myasthenia gravis. Myasthenia gravis is a chronic autoimmune disorder that is caused by breakdown in communication between the nerves and muscle, which leads to droopy eyelids, difficulty swallowing, sometimes double vision, and unsteady walk due to weakness and atrophy in the muscles. And a lot of times the condition worsens after long periods of activity, and when people rest up, their situation or condition improves. There is no known treatment for myasthenia gravis. There's no known cure for myasthenia gravis, but a lot of treatments are present to help manage the condition. Okay, so acetylcholine and acetylcholine receptors are very, very important at the neuromuscular junction. Once muscle contracts, the muscle needs to relax again. Okay, because if the muscle remains contracted, it will cause a constant constriction in like your bronchial muscles. It will cause paralysis, and possibly it can cause death if you cannot relax your, say, muscles that you're required to breathe, right? So once acetylcholine has been used for reception of the nerve impulse, it needs to be broken down. Okay, this is called acetylcholine inhibition. Okay, and how is acetylcholine broken down? It is broken down by an enzyme known as acetylcholine esterase. Acetylcholine esterase breaks down acetylcholine to allow relaxation of the muscle. Okay, and if this acetylcholine esterase is blocked, the concentration of acetylcholine will remain constantly high, and it will cause extreme, say, bronchoconstriction, and paralysis. And if you have your bronchoconstriction that is irreversible, it will lead to death, because you won't be able to breathe, because all your diaphragm and the bronchial muscles will remain contracted, right? And this is what happens in nerve gas, like sarin gas, where people inhale the sarin gas and it causes the bronchial muscles to remain contracted, because the sarin gas blocks acetylcholine esterase. You see all these neuromuscular junctions. These are all individual muscle fiber, and this is a synaptic knob, and these are the nerve fibers at the neuromuscular junction that communicate with the muscles and tell them what to do. So this is the muscle, this is the synaptic knob, and there is a little gap in between the two known as the synaptic cleft, and all these lines that are highlighted in here in blue are the nerves, okay? And this is again the synaptic knob that is highlighted. So all muscle cells and nerve cells are excitable, meaning they can respond to a stimulus. A stimulus is caused by a disturbance in the electrical activity and the electrical charges inside of the cell. Okay? At rest, the cells have something called as resting membrane potential. And the study of the electrical activity inside of the cell is known as electrophysiology. And like I said, all this is due to the charge inside of the cell, and this is determined in terms of voltage. So the voltage or electrical potential is a difference in electrical charge in a cell from one point to the other. Okay, and at rest, these cells have a resting membrane potential, and this is about minus 90 millivolt to minus 95 millivolt in skeletal muscle cells. And the main pump that regulates the electrical potential or the membrane potential is the sodium-potassium pump. If you remember from the third chapter, the sodium-potassium pump moves in 3 sodium ions out of the cell for every 2 potassium ions it puts into the cell. So this causes the inside of the cell to become more negative relative to the outside of the cell. So here, let's just assume this is the inside of the cell, and too many sodium ions are moved out and too little potassium ions are brought in. It causes the inside of the cell to become more negative than the outside. So it gives the cell a negative charge. That's how a cell has a negative resting membrane potential. Okay. Now an action potential, let's write it down. An action potential an action potential occurs when a nerve sends information down the axon away from the cell body of the neuron. And this causes an explosion of electrical activity, which is created by the disturbance in electrical charges. And this is called a depolarizing current. So an action potential is caused by a depolarizing current. This means that some event causes the resting potential to move from a negative voltage towards a positive voltage, towards 0 millivolt. Okay, so this event of explosion of electrical charges is called an action potential, or it is called a spike or an electrical impulse as well. So in physiology or in neuroscience, you can use the words such as action potential, spike or impulse, or depolarization. This all means the same thing. When the action potential reaches the end of the axon terminal, it causes depolarization due to disturbance in the electrical charges. This causes the sodium channels and the calcium channels to open up, and there's an influx of sodium and calcium ions into the synaptic knob. Okay, and this activation of the sodium channels due to depolarization of the axon terminal is called voltage gated pump activation. So it causes the voltage gated sodium pumps to open, which leads to an influx of sodium ions into the synaptic knob. This change in the electrical conductivity causes the activation of calcium ions, which leads to influx of calcium ions into the postsynaptic knob. Now, at the synaptic knob, there are tiny vesicles containing the neurotransmitter. This neurotransmitter, if you remember, is called acetylcholine. Acetylcholine. Acetylcholine is the neurotransmitter which merely transmits the message from the nerve to the muscle. Alright, so for these acetylcholine vesicles to be activated, calcium must bind to them. So when calcium enters the cell, these vesicles get activated by binding of calcium ions. And these vesicles fuse with the plasma membrane, and the acetylcholine gets released into the synaptic cleft. This is the synaptic cleft, which is the gap in between the two cells. And this process of release is known as exocytosis. Exocytosis. So in chapter 3, if you remember, we studied about the mechanisms of things entering and leaving the cell. Acetylcholine leaves the axon terminal by the process of exocytosis. Alright, so now let's look at the muscle cell, what's happening over there. How does this acetylcholine molecule activate the muscle cell? The muscle cell has a whole bunch of ion channels, and they are embedded either into the plasma membrane of the muscle cell, or they sit right on the surface of the muscle cells. Okay, so the first channels that we are going to talk about are the sodium channels. These sodium channels sit very snugly with receptors for acetylcholine, and these receptors are simply called acetylcholine receptor molecules. Okay, these acetylcholine molecules bind to the receptor on the surface of the muscle cells, and they in turn activate the sodium channels. The sodium channels open up, and there's an influx of sodium ions into the muscle cell. Now there's a whole bunch of them on the surface, but for ease we're just going to mark up 2 of them. When there's an influx of sodium ions into the muscle cells, this is a muscle cell, it disturbs the electrical activity inside the muscle cell, and now it causes opening of voltage-gated calcium ions. So these are the voltage-gated calcium ions, which causes an influx of calcium ions into the muscle cell. Okay, now let's quickly go over what's happening over here. The action potential arrives at the axon terminal. Let me look at the laser pointer quickly. The action potential arrives at the axon terminal. It causes an influx of sodium and calcium ions into the synaptic knob. The acetylcholine vesicles get activated by binding of the calcium ions. The vesicles fuse with the plasma membrane of the axon terminal and release the acetylcholine molecules by exocytosis. The acetylcholine molecules bind to the receptors of acetylcholine on the surface of the muscle cells. These acetylcholine receptors sit snugly with sodium channels. So when acetylcholine binds to the receptor, they activate the sodium channel, which causes an influx of sodium ions into the muscle cell. This influx of sodium ions causes an activation of voltage-gated calcium ions. So these are voltage-gated calcium ions. I should write that down real quick. These are voltage-gated calcium channels. Voltage-gated calcium channels. Okay. So inside of the muscle cells, there are specialized structures known as sarcoplasmic reticulum or SR. The sarcoplasmic reticulum stores calcium ions. So calcium ions are stored right in here. So for sarcoplasmic reticulum to release calcium ions, it must bind to calcium ions. Okay, so these influx of calcium ions are actually useful to release calcium ions from the sarcoplasmic reticulum. So when we have enough calcium ions inside the muscle cell, these calcium ions bind to receptors on the sarcoplasmic reticulum, and the sarcoplasmic reticulum causes the release of, I deleted the sarcoplasmic reticulum. The sarcoplasmic reticulum opens up some channels and it releases a whole bunch of calcium ions into the muscle cell. Okay, so this step is called calcium-induced calcium release. Okay. So this is how calcium finally gets released into the muscle cell and binds to the troponin, and the muscle contraction begins. Okay. So let's quickly revise what we looked at over here. The first step is arrival of the action potential at the axon terminal. There's an influx of sodium and calcium ions into the axon terminal. Calcium binds to the acetylcholine vesicles to activate the acetylcholine vesicles. Acetylcholine gets released into the synaptic cleft by the process known as exocytosis. The acetylcholine bind to the receptors on the muscle cell, which in turn activate the sodium channels. There's an influx of sodium ions into the muscle cell, which disturbs the electrical activity inside the cell. This activates voltage-gated calcium channels and causes an influx of calcium ions into the muscle cell. And this influx of calcium ions then activates the sarcoplasmic reticulum. The sarcoplasmic reticulum releases a lot more calcium ions into the muscle cell. This is called calcium-induced calcium release from the sarcoplasmic reticulum. This released calcium ions attach to the troponin, which is on the actin. Troponin changes conformation, opens up the binding site, and allows myosin head to bind to actin and causes contraction.